morning, everyone. We are very pleased to have Dr. Jeffrey Holt from Boston Children's Harvard Medical School to join us today. Dr. Holt is a professor of otolaryngology, head and neck surgery, and neurology at Harvard Medical School. He earned his doctorate in physiology from University of Rochester. He then completed his postdoctoral fellowship at Harvard Medical School. He joined the Department of Otolaryngology and Neurology in Boston Children's in 2011. Dr. Holt serves as one of the two principal investigators of the Holt-Gallup lab. His lab is very well known for studying molecules and mechanisms of sensory transduction in inner ear hair cells and developing novel therapeutic interventions for genetic hearing loss. This year also, Dr. Holt was elected as a member of National Academy of Science, which is one of the highest honors in science. Dr. Holt, again, congratulations, and thank you for accepting the invitation to give a talk today. Thank you so much.
Wonderful. Thank you, Armine. It's very nice to be with you all this morning, and an honor, really, to be joining you at the House Institute. I've never been there myself, so I guess virtually is a good opportunity, but I hope someday to visit in person. So I want to jump right in and tell you about some of the gene therapies that we've been thinking about for genetic hearing loss. First, I should at least flash up my disclosures, which you can see here. And so I'll begin with the numbers. You're probably familiar with some of these numbers, but just so they're fresh in all of our minds. The World Health Organization currently estimates there are 430 million individuals with hearing loss. Now, unfortunately, the situation's only going to get worse, such that by the year 2050, just 25 years from now, that number is expected to grow to 2.5 billion. That's with a B. A lot of zeros here. And unfortunately, I've got one more zero for you. The number of FDA-approved medicines is currently 0. Now, you've probably also heard that may be changing. Last year, for the first time, we now have a gene therapy for a genetic hearing loss. This made the news, of course. It was all over in the New York Times. This is Assam, a young boy who was injected with a gene therapy and recovered some auditory function. Quite remarkable. This made it into the news. You've probably seen it yourselves. A number of different news stories, NPR, ABC. Here was one of my favorite news stories. You all in Los Angeles, I'm sure, can appreciate this one. Here was the breakthrough news for hearing loss, but I liked it because it made top billing above the Oscar nominations. I feel like our field is really making progress if we can make that kind of a headline. So for me personally, this story began about 25 years ago. When I was a postdoc, we began thinking about using viral vectors to put DNA into hair cells. And this was our first publication in this area, where we used a viral vector to deliver the coding sequence for green fluorescent protein, and showed that we could target the sensory hair cells of the inner ear and light them up with a marker glowing green. So this was a proof of concept that this could be done. And we took it one step further. Instead of just turning cells green, we used a functional assay where I linked the green fluorescent protein to an ion channel known as KIR 2.1. And here you can see that restricted the localization of the green fluorescence to the membrane of the cell. But then we could also record the currents, the electrical currents, in a control non-transfected cell. This was the family of potassium currents that were evoked. But in a transfected cell that was GFP positive, we got these large inward potassium currents, inward rectifier potassium currents. So this was the first case of a viral vector being used to change the function of a sensory hair cell. And it was really a eureka moment and provided, I think, some of the first insight that viral vectors could one day be used to alter the function, perhaps improve the function of sensory hair cells. And so 25 years later, I'm not going to review the entire field, but 25 years later, we now have the first gene therapy for a gene known as otoferlin. And so you've probably heard about this as well. Otoferlin is involved in synaptic transmission. The coding sequence is too large to fit into a single AAV, so a dual vector strategy was developed, injected into the ears of patients. The dual vectors can promote assembly of this DNA sequence to allow generation of a functional protein and recovery of function. One of the remarkable things, I think, is that there are now 5 different companies that are pursuing this, and this is one of the rare cases in science that you've got consensus in the field amongst a group of scientists and amongst the funders who have established these strategies. And the great news is that it does seem to be working. You know, I think there's over 20 patients who have been injected so far. Maybe the count is up to more than that at this point. We've got some of the first cases being injected at Boston Children's later this month. So coming back to the numbers, I want to ask the question, well, what next? If we've got now proof of concept that this can work for otoferlin patients, what's going to be next? And so the prevalence of otoferlin is around 10 to 20,000 in the US, somewhere in that range. That corresponds to about 400,000 worldwide. And so that's a significant population, but it's still just a fraction of the 430 million who are out there. You know, that's 0.1% of the population. So, you know, while it's exciting to see that this seems to be working, it's clear that we have a lot of work to do if we're going to begin to get even close to addressing this huge patient population in need. So if this is 0.1% of the patient population, what else? That's the question I want to spend the rest of my time here today addressing. What might be next? What other targets? There's at least 100 different forms of genetic hearing loss. And I want to tell you about one of them that we've been thinking about. It's known as TMC1, which stands for transmembrane channel-like 1. And this one has, I think, a similar incidence and prevalence as otoferlin. We estimate there's about 250 patients worldwide, maybe around 10,000 in the US. That corresponds to about 132 new cases per year, 132 new babies born with TMC1 mutations. Now that's in the US. In some populations, it's more prevalent. In the Middle East, for example, it's the second most common form after GJB2. But within the US population, we've further broken that down. The 132 patients, there's about 82 who carry recessive loss of function mutations, and about 50, we estimate, that are carrying dominant mutations. And so the TMC1 gene, let me back up and spend a little bit of time talking about what this is and what it does. It's involved in the sensory hair cells of the ear and involved in the sensory transduction process. I'm sure you all are aware that hair cells have the job of converting a mechanical stimulus into an electrical signal. The TMC1 protein is found here at the tips of the sensory hair bundles where it's stained in red. You can see it here in this punctate position. And so we think it's involved in the sensory transduction process, which of course, if it's there, it must be right there at the tips where we've shown it. And when these bundles wiggle back and forth, they're converting this mechanical stimulus into an electrical signal that we can record in the lab electrophysiologically. And so the model you might also be familiar with suggests that these mechanosensitive channels here at the tips, when the bundle swings back and forth, these will open and close. And when they're open, cations flowing into the cell, primarily calcium and potassium, will evoke that electrical response. Now, over the past 15 years or so, many of these elements have been identified at the molecular level. This extracellular filament, known as the tip link, is composed of cadherin 23 and protocadherin 15. There's a motor molecule that adjusts tension within this structure that's composed of myosin 7A. Interestingly, these are all Usher syndrome genes, so mutations in these cause both deafness and blindness. The ion channel itself has been the focus of major attention over the past really 40 years, since it was first characterized by David Corey and Jim Hudspeth in the late 70s and early 1980s, and has been a big question mark in the field. But we think that we now have the definitive evidence suggesting that this channel is formed by the TMC1 protein. And I'm not going to review all of the evidence, but a few key bits of evidence that we published a few years ago in this manuscript, which appeared in Neuron. The idea here was to use a chemical genetic strategy, introduce mutations into the TMC1 protein that would sensitize that protein to a genetic inhibitor. And so the inhibitor was called MTSET, and the experiment was to deliver a series of repeating hair bundle deflections, apply the drug, and then look for changes in function. And so here's what the data actually look like. Nice looking transduction currents, but as soon as the drug hits, you could see a decline in the amplitude of this response, and it does not wash out at the end. This was very robust and repeatable. Here's a second round where you can see the same effect in another cell. And then a couple of important control experiments where we introduce a TMC1 mutation, but use a saline puff without the inhibitor. And in that case, you do not see a decline in the response. And in another experiment with a wild type cell that doesn't carry the mutation, but with the inhibitor, and we don't see a decline there either. So it really depends on the chemical and genetic mutation being present at the same time, the intersection of the two to get this effect. And this occurring in real time was very strong evidence. And so we did this in 17 different genetic mutations that we introduced into sensory hair cells. And the data are summarized here. We found 5 different locations within the TMC1 sequence where we saw a significant reduction in the amplitude of the current. And what this told us was that these particular locations seem to be lining the ion channel pore because they're affected by application of the drug. Whereas these other sites where there was no change probably were not part of the permeation pathway. A second assay was to look at calcium permeability, and this is a busy slide. I'm not going to go through all of these data, but I can summarize by saying that we found 11 sites that were affected either by the genetic mutation or the combination of applying the drug together with that genetic mutation. So these are really hallmarks of an ion channel, the current that flows through it, as well as the kinds of ions that flow through. So affecting both current amplitude and calcium selectivity was strong evidence that the TMC1 protein is indeed forming the ion channel in sensory hair cells. And so stepping forward again, we now have a pretty clear picture of what this ion channel looks like. This is a cryo-EM structure that was generated by a colleague of mine, Eric Guo's lab. And so the TMC1 protein we think is a dimer. I'm going to put this in motion. This is kind of spinning. This is the outside of the cell. This would be the inside of the cell. And you can see the 2 dimers. One half is here. This is the other half. There is a permeation pathway, interestingly, in each dimer, one on this side and one on this side. And it's composed of 10 transmembrane domains. Now, this is important information for understanding the structure and the function of the ion channel, but it also gives us insight into why genetic mutations lead to hearing loss. And we now know of over 100 different mutations that are affecting the TMC1 protein. So this is the DNA structure of the TMC1 gene, and most of these are colored in black, indicating that these are recessive loss of function mutations. And so you would need to carry 2 copies, one inherited from mom, one from dad, to have a genetic hearing loss. About 10 of these are dominant mutations, so those are pictured here in red. And so a single copy, together with a wild type copy in a heterozygous situation, could cause a dominant progressive hearing loss. And so we wanted to then ask, well, can we develop a gene therapy strategy to target some of these genetic mutations? And so we've explored two general strategies. One is gene replacement and one is gene disruption. I'll first talk about gene replacement. And the idea here is that if you've got a genetic mutation that leads to a non-functional protein and a disease phenotype, well, maybe with a gene therapy where you deliver the wild type healthy DNA sequence into the cell, let the cell generate a functional protein that replaces the absent or non-functional protein, you can correct that disease phenotype. And so we've done this a number of times over the past 10 years and kind of improved our strategy over time. And I'd like to walk you through some of those data. One of the innovations was to identify viral vectors that are particularly efficient for targeting the sensory hair cells of the inner ear. And we've landed on one recently in the past 5 years that we're particularly fond of because it's got really high efficiency. We can target almost 100% of the inner hair cells and about 95% of the outer hair cells. And you can see that even better in a higher magnification view where all of the inners are targeted and most of the outers as well. This is a synthetic AAV. It's known as AAV9 PHPB, and it's our current favorite vector for delivering into the sensory cells. And so instead of just turning cells green, again, the idea is to introduce the wild type TMC1 gene sequence into a TMC mutant mouse that lacks function. So these are measurements of hair cell transduction where we deflect the hair bundle, measure the response, and in a mutant animal, we have flat line responses. So we get no sensory transduction. But after delivering TMC1, we see that we can restore function in outer hair cells at postnatal day 7, inner hair cells at postnatal day 9, and this is durable postnatal day 30, latest time point we've recorded. The stimulus response relationship is similar to those of wild types, and so that's quite encouraging. So we're able to restore function at the apex of the cell, the sensory hair bundle. And of course, for sensory information to be transmitted, we need to have normal function at the synapse. So we've also looked at the synapses of these cells. In wild type cells, we've quantified the number of synapses at the base of inner hair cells. We actually find there's a deficit or a loss of synapses in the TMC mutant animals. But if we quantify these, normal synapses are shown here in black. And in the mutant animals, you can see a significant reduction. But after reintroduction of TMC1 into the mutant animals, we find that we can restore the number of synapses and hence transmission of sensory information into the postsynaptic 8th cranial nerve neurons. To look at whole cochlea function, we record auditory brainstem responses. And here are traces from animals that lack TMC1. And so you can see the flat lines indicating profound deafness. A wild type family of traces is shown here on the right. And so here are thresholds of about 30 decibels in this animal. After we introduce our TMC1 gene therapy into the mutant animals, we find that we can restore those thresholds, in some cases with thresholds equivalent to those of wild type. So this is also encouraging, but again, we wanted to assay at multiple levels in the auditory system. And so in collaboration with Dan Pauly's lab, which is just across town at Mass Eye and Ear, we have recorded the responses in auditory cortex. So in this case, we inject one ear, play sounds into this ear, and then record the responses in the contralateral auditory cortex. And these are raster plots showing action potential firing from neurons in auditory cortex with louder and louder sounds on this axis. This is a control animal, and you can see evoked responses in response to this tone pip. right here tend to cluster. In a deaf animal, we introduce a tone pip and we do not evoke those responses. But after injection of our gene therapy into the inner ear, we see that we can begin to evoke those responses again. And so Dan's lab recorded from over 400 cortical neurons, and the wild types are shown here in gray. A deaf injected animal, these responses are shown in green. And in this particular animal, you see that they cluster around the 8 kilohertz frequency range with thresholds, some of them best thresholds around 30 to 40 decibels. The shape of these curves was very similar to those of the wild types, suggesting that the tuning properties of the cortical neurons are similar. So that was also encouraging. Next question was, well, what about behaviorally? Unlike humans, in a mouse, you can't assay this too easily, but we can rely on the auditory startle response. So we'll play a sudden loud sound to a mouse. A deaf mouse hears nothing and doesn't really respond to that. A wild-type mouse will jump in response to a sudden loud sound. We actually can measure the amplitude of that jump. And so we've quantified that here. Here's the wild-type response in black. The deaf animals with louder and louder sounds do not respond at all, but the deaf injected animals respond again, similar to those of the wild type. And so we've been able to show then that with a single injection into the inner ear of these deaf animals, we can restore sensory transduction, synaptic transmission, whole cochlea function in terms of the ABRs, responses of cortical neurons, and these behavioral responses. And so we think that bodes well for TMC1 gene therapy. Historically, going back to our first data set, we did this in 2015. And when we first published this, we got responses that were improved compared to the profoundly deaf animals. And this actually stirred up a bit of attention. I was interviewed on NPR, it was on the BBC, and my basic science colleagues were very excited to see this. But when I showed it to my clinical colleagues, they were not too impressed. They said, you know, Jeff, if you tried to do this in a patient, this is going to be useless. These are still going to be candidates for cochlear implants. And, you know, so that's when we went back to the drawing board. We tried to iterate on this, improve our strategy, and we found viral vectors that had better efficiency. In 2019, we published this level of recovery, which was better, but we went on further to identify other viral vectors that improved it further. We changed the promoter that was used. We changed the coding sequence. We used a human codon optimized, and now we're to the point where we can get robust responses equivalent to wild type across the entire auditory spectrum. And so we're really encouraged by that. And this is the actual construct that we hope that we can get into the clinic in the coming years. And so this has been the strategy, the gene replacement strategy for restoring function in recessive hearing loss. And so we think that may account for the majority of the cases. All of those shown here in black could potentially benefit from a gene replacement type strategy. We tried it on those shown in red. These are the dominant mutations. And we thought, well, maybe if we overexpress the wild type sequence, maybe we can compensate for this dominant mutation. But in fact, that did not work. Unfortunately, the dominant mutation was too strong and just could not be overcome with a wild type sequence. And so we went back and looked at this other strategy. Whoops, where did it go? There we go, which was gene disruption. And the thinking here is if you've got a heterozygous situation where you've got one dominant allele and one wild type allele, well, maybe you can use a gene silencer, knock down the dominant allele, get rid of that, leave the wild type intact, and then allow for correction of that disease phenotype. And so we decided to do that using a CRISPR-Cas approach. And we've done that on a few different occasions, trying to improve upon that. And let me walk you through some of those data. We looked at a TMC dominant mutation known as Beethoven, named after the classical music composer Ludwig van Beethoven. The mouse model here carries a single point mutation that leads to an amino acid substitution. And so again, the thinking is, well, if we can get rid of this dominant allele, leave the wild type allele intact in a heterozygote, maybe we can overcome this. And so we developed a dual vector approach where we carry the Cas9 enzyme, one of the components of a CRISPR gene editing approach, that's packaged into one vector. And then in the other vector, one of the key elements is this guide RNA right here. The guide RNA needs to be selective so that it recognizes the mutant allele, but does not bind to this wild type allele. And we also carried green fluorescent protein just as a marker to see which cells had taken up the vector. So we screened a number of different guide RNAs, and I show a few of those sequences right here. And again, we wanted to find one that was selective for the mutant allele, but not the wild type. And so here are the data. This is an example, one of the guide RNAs that was highly efficient, but unfortunately this one was targeting both alleles. We got significant disruption of both the wild type and the mutant. And so that wasn't going to be useful. That was going to lead to deafness itself. But after screening a bunch of these, we found one that had lower efficiency for the wild type allele and higher efficiency for the mutant allele, up to about 60%. And so we decided to package this guide RNA into our dual vector approach and inject that into the ears of these Beethoven mice. And so our first assay was to look at the hair bundles. And so wild type hair bundles you can see here from uninjected animals. Compared to the Beethoven, these you can see you don't have to be an expert in hair bundle morphology. These are just badly disorganized and non-functional. But if we injected these early in development, we found that we could preserve this morphology, which you can see here in higher magnification. So the nice conserved hair bundle morphology, more similar to those of the wild type. When we looked at the function of this, we found that the Beethoven animals had a progressive hearing loss. These are auditory thresholds, ABR thresholds, over 4, 8, 12, and 24 weeks of age. The wild type animals have stable thresholds, but if we injected the Beethoven animals with our dual vector approach, we found that we could preserve those thresholds and have stable auditory function. And we've now gone out to 1 year of age, suggesting that we were able to selectively target the mutant allele and leave the wild type allele intact. And so that was encouraging. The challenge, though, is that that particular strategy was for just one of these dominant alleles, the M418K, and is not going to be effective on the others. And if you're really going to develop this as a therapeutic that's going to get into the clinic, you would have to have a selective strategy for each one of these different dominant alleles. And so you end up with 10 different therapeutics, 10 different clinical trials, and the numbers of patients and the time and the expense involved begins to become prohibitive. And so we thought, well, wouldn't it be great if we could develop a strategy that could address all of these mutations, both the recessive mutations and the dominant mutations, with a single strategy? And so we went back and thought, well, maybe we could combine these, the gene replacement and the gene disruption, into a single strategy, which we've decided to call the disrupt and replace strategy. And the thinking here, this is unpublished work, but I'd like to tell you about this, what we're currently working on, developed by a couple of postdocs in the lab. This is a dual vector approach where one of the vectors is similar to the disrupt strategy I already mentioned. It carries this SP-Cas9 enzyme. The other one carries a guide RNA, but instead of GFP, we can replace that with the TMC1 sequence. This is the wild type sequence. And so together, we may be able to disrupt and then replace with TMC1. And so in this case, we need to use a guide RNA that actually targets both of the endogenous alleles. We're going to get rid of the mutant allele and the wild type allele. And you may recall, I showed you these data earlier. This first one right here actually has pretty high efficiency for disrupting both the endogenous wild type allele and the mutant. So we can get rid of all of them, everything that's there with this strategy, and replace it with this TMC1 sequence. Now, we also had to take this a step further and make sure that the guide RNA that we're using doesn't disrupt our exogenous TMC1 sequence. So we used a synonymous change in the TMC1 DNA sequence so that the guide doesn't recognize this TMC1 sequence, but we made sure that the synonymous change preserved the amino acid sequence so that the protein would still have function. So to summarize, the disrupt and replace strategy is going to use CRISPR-Cas to get rid of the native TMC1 alleles, both dominant and recessive, and replace it with this wild type TMC1 sequence that is insensitive to the disrupt strategy. So we've injected that into animals that carry a dominant mutation. This one is known as D569N. These animals have reduced mechanotransduction currents. We find that we can restore the amplitude of those currents. Preliminary data here, we've done ABRs, and this is a busy slide, but let me draw your attention to these. We've got recovery of function in many of these animals, which are shown here in green. Not quite back to the wild type level yet, but we're working to improve the efficiency of that. That's in a case where we've injected a mouse that carries the dominant mutation. We've also done the same sort of thing in mice that carry a recessive mutation, and we're getting recovery of function there as well. So we were encouraged by this, and we're thinking that this strategy could be used in the clinic eventually to treat patients who carry both dominant and recessive TMC1 mutations. And so that's kind of where we are right now. And so to conclude this first part, we've identified viral vectors. Our favorite is known as AAV9-PHPB that can target 95 to 100% of sensory hair cells. The gene replacement strategies we've shown we can recover function back to wild type levels across the entire auditory spectrum. The disrupt strategy with CRISPR-Cas seems effective for dominant mutations, but my favorite, which I hope we can eventually get into the clinic, is this disrupt and replace strategy, which could potentially target all of the TMC1 mutations. Now, next steps really are, I think, this gene replacement strategy is closer to clinical application. I'm hoping within the next couple of years, we could get that into clinical application. And so following the otoferlin success, we're hoping that TMC1 gene replacement may be coming down the line within a couple of years. And our next steps are to begin generation of the clinical grade material. We're working on that right now. We need to do the farm tox assays, make sure that we're not having any deleterious effects with this approach. And then we plan to submit this for an FDA application for an investigational new drug within the coming year, year and a half, and then hopefully shortly thereafter initiate clinical trials for TMC1 gene therapy. So let's see, I think I've got a little more time. So in the next part, I would like to tell you about another gene therapy that we're also working on. This one is for another form of genetic deafness, a gene known as stereocylin, STRC. STRC is in the US the second most common form of genetic hearing loss, accounting for 16%, where GJB2 is in the 25 to 30% of genetic hearing loss. This one is probably a 16%, accounting for maybe 100,000 patients in the US. About 1,200 babies per year, we estimate. That actually corresponds to about 2.3 million people worldwide with STRC mutation, so it's a significant number. This protein, the stereosilin protein, is present at the tips of sensory hair bundles. It actually forms the top connectors, which connect to the overlying tectorial membrane, as well as these adjacent top connectors linking stereocilia. You can see it here labeled with an immunogold. So we generated a mouse model of this. This was a gene knockout, and we were able to confirm the knockout of that gene. We found that, interestingly, the hair bundles still function normally, so mechanotransduction currents were just fine. The animals had elevated distortion product emissions. Interestingly, this one's expressed just in outer hair cells and not in inner hair cells. So we saw elevated DPOAEs and elevated ABRs as a consequence. The challenge here is this one's also too big to fit within a single AAV, which has a packaging capacity of 4.5. This is about 6.3 kb. And so we had to figure out how to split this structure in two. And we screened a number of different strategies. I'm not going to go through all of these data, but instead we found one, this variant right here, we were able to split the sequence into and then use a protein recombination. So unlike the otoferlin strategy where you have DNA recombination, we actually do this at the protein level and found that there was a greater efficiency for having protein recombination and restoration. And so here's a Western blot showing that we can get recombination of that protein. And so we packaged that and injected those into mice that carry the stereosilin mutation. This is the protein antibody localization where you see it at the tips of outer hair cells. The knockout mice have no localization of the protein, but if we injected the dual vector approach, we found that we could get the protein expressed in about 63% of outer hair cells. So it's not quite as efficient because of this dual vector approach, and we're still trying to optimize this further. But you could see recovery of protein expression in 63% of those. When we looked at scanning electron micrographs, you can see wild type structures here. The STRC knockout mice, they've got slightly disorganized bundles, not as bad as some of the TMCs, but you can see higher magnification views. They're a little bit disorganized. And interestingly, in the injected animals, you can actually see this side by side. I really liked this image because here's one that presumably did not take up the dual vectors, and one neighboring cell that did take up dual vectors, you can see with restored function. And if we zoom in even further, you can see a pair side by side, one that was not expressing stereosilin with one that does. And if you look closely, you can even see the protein product of the gene therapy. Right there are those side links that are formed by the stereosilin protein. Functionally, because this is expressed in outer hair cells, we wanted to look at the distortion product otoacoustic emission. And so as you may be aware, if you play two tones into the ear, you can record this distortion product, which if we amplify the signal here, you can see these distortion products right here. The stereosilin knockout mice lack those distortion products, but after injecting with the dual vectors, we find that indeed we can restore the DPOAE response. And here it is as a function of frequency. And so there was a bit of a scatter here in the responses. We injected 20 animals, and the mean of all 20 is shown in green. I think some of that scatter is, again, because of the dual vector. Some cells took up just one vector, some took up the other. Those where we had good responses, I think, had high efficiency and took up both vectors, and the mean of those are shown in purple. And so some of the best cases, we were getting closer to wild type responses. Likewise, with the ABRs, we were able to get some very good responses in a subset of cells, which are shown right here. And so we're still working to optimize that dual vector efficiency even further. And so to summarize the last part of the talk here, stereosilin, we think, is the second most common form of genetic hearing loss, at least in the US. The mice that we generated are a good model for this. And so using the dual vector approach, we can restore protein function, we can restore DPOAEs, hair bundle morphology, and ultimately the ABRs. And so we think that this one can be further developed and could potentially benefit up to 2.3 million people worldwide. And so, that's kind of where we are right now. I do hope that we can get some of these strategies into the clinic in the coming years. I think probably TMC1 gene therapy will be next, perhaps the next one after otoferlin, and maybe stereosilin will be a little bit further, but hopefully not too far down the road. And so just in conclusion, this is my group here in the lab. My longtime collaborator, Gwen Jeliak. Some of you might recognize, this is Jonathan Ashmore, who is not a member of my lab, but showed up to visit one day. And so this is the rest of the team, and I'll stop now.